that we're talking about purification. So you can purify your protein using chromatography, where we said you can use gel filtration, ion chromatography, depending on a particular property of the protein you feel like you can explore. Or you can decide to even do all of them, so that stepwise. So what happens is when you are doing this, for example, let me give an example. You are dealing with a protein that is an enzyme. So we decide to be sure that, yes, what you are dealing with contain the enzyme you are talking about, you are working with, or you are interested in. So right from beginning, when you prepare the homogenate, after getting the homogenate, after disrupting the organelle, you can check the enzyme activity of the homogenate. You can see the enzyme activity. That's tell you that, yes, that enzyme I'm looking for is there. So after doing the precipitation, you precipitate, you take it out, you now do the purification. For example, let's say you started with gel filtration, where you prepare your own stationary phase, where the stationary phase containing these substances that can allow the movement of smaller, and then retaining the bigger ones, and use the buffer to wash them down. So as you are collecting the fragment, you will say, maybe after so minute, I will collect this fragment in the first test tube. After so minute, the next fragment, like this, like this. After you finish this, then you can now check each of these test tubes, the activity of your enzyme, which one have the activity of your enzyme, if it is an enzyme. Remember, I'm giving it an example. So you will definitely find some of them. If your protein is still there, you find some of those ones with activity, just maybe. So you can have, for example, let's say, based on this image, we can say test tube 3 and 4 have some proteins that are your enzymes. Of course, you can have 5, 6, 7, 8, 9, up to 10. They have a protein, but they don't have the enzyme activity, which simply says they are protein, but they are not the protein of your interest, the protein you are wanted to isolate. Then you have 3, 4, maybe and 5 that have some activity. And that doesn't mean it's only your enzyme that is there. So you can still subject it to the next, let's say, cation exchange. You can do that and collect fractions from the fraction. You further check the activity. You can also go for affinity like this, at least by 1 or 2, even enough. So you can have some fractions which have the activity and tells you that it is your enzyme. So you can pull them. When you pull them, now the next thing is you can go for electrophoresis. You can go for electrophoresis. In electrophoresis, you separate them on the basis of charge. You separate your protein on the basis of charge. So the essence of electrophoresis is more or less a confirmation of the protein you are working with. First of all, is it a mixture of protein? Is what you have, is it only your protein that is there? What is the degree of purity of the protein? And you can even determine the isoelectric point and the molecular weight of the protein. What you simply need to do is you just take your sample and load it into your electrophoretic gel, and then run the gel. When you run the gel, you can have, for example, you can even run it together with a sample that you know the content of the protein. For example, let's say you are looking for at least you are seeing this image. You are seeing the one at the right, where we have the standard and we have unknown. Can you see it, Mariam? Can you see it, Mariam?
Yes.
So for example, let's say the enzyme you're interested in, you can see carbonic anhydrase, and the molecular weight of the carbonic anhydrase is there. So it is carbonic anhydrase. So you have a standard containing a number of proteins. We can see the number of proteins and their different molecular weight. We can see myosin, beta-galactosidase, glycogen phosphorylase B, bovine serum albumin, ovalbumin, carbonic anhydrase, soybean trypsin inhibitor, lysozyme, and their various molecular weights. So you can see the bond forming after separating using the gel filtration, using electrophoresis. So the next one, which indicates 2 here, the well 2, what you added is your sample, which you don't know the protein, and you wanted to isolate carbonic anhydrous. You want to be sure it is carbonic anhydrous. So when you run it together with the standard, where the standard you have carbonic anhydrous, you are expecting your own protein under well 2 to be somewhere situated around the carbonic anhydrous. Is that clear? That tells you that, yes, what you have is exactly carbonic anhydrous. But you can have some band which tell you that apart from carbonic anhydrous, you have other things also. So if that thing also is part of your standard, you will see what it is. If it is not part of your standard, you will just know the molecular weight of the protein. At least you have an idea, the molecular weight of the protein. You can even find out from which proteins have that molecular weight. So their possibility is one of them, although it can vary. Are we clear? At least we've known how to isolate a protein now. Mariam. By now we have an idea how to isolate a protein? Class?
Yes, sir.
Good, Helen. So the next thing is, how do you determine the composition, the amino acid composition of the protein? Now, just imagine you have your own protein, you isolated at the end, you run electrophoresis, you identify the protein. This protein is carbonic anhydrous. Now, how do you know the amino acid composition? Which amino acid form this protein? What amino acid? So you can determine. How do you determine? First of all, you break the peptide bond. Remember, the protein contains the amino acid linked by the peptide bond. Now you break all the peptide bond. How do you break the peptide bond? The first step, we call it hydrolysis. So you hydrolyze the protein using a strong acid at high temperature for about 24 hours to 74 hours. So after subjecting it to this condition, highly acidic condition of 6 normality, HCl, at 110 degrees centigrade for 24 hours to 72 hours, you have all the peptide bond broken. So what you have all your amino acids. What do you do? The next thing now is to use chromatography to separate them. The most common one you can use cation exchange chromatography, a form of ion exchange chromatography. So you separate them based on the charge. When you separate them based on the charge, you have each amino acid elutive coming out of the column individually. So each amino acid coming out, you can collect it. You can use spectrophotometry to detect it. How do you determine a amino acid? Spectrophotometry. One of the methods we use in the lab, we use ninhydrin reaction. That this can react with ninhydrin. An amino acid can react with ninhydrin. If it reacts with ninhydrin, it forms a color derivative. It will form a compound that is colored. So usually all the amino acids, when they react, they form a purple or blue color, and they absorb at 570 nanometer. So first of all, when it reacts with an anhydride, you know, yes, you have an amino acid inside. You are sure of the amino acid inside. The next thing is you determine the concentration of the amino acid. How do you determine the concentration of the amino acid? You will do, you take the absorption at 570 nanometer, and that will tell you the concentration of the amino acid that is there. So also, in the case of some amino acid like proline, which when react with anhydride, it gives a yellow color instead of the purple or blue, it can be measured at 440 nanometer. So this determination usually, this method is usually incorporated in form of what we call amino acid analyzer. So you have a machine called amino acid analyzer that contain, after hydrolyzing your protein, you can put the protein suspension or solution inside the amino acid analyzer. The amino acid analyzer contains the ion exchange chromatography, or in form of HPLC, that will separate and then detect and then analyze individual amino acid. And it will tell you that this protein contains so number of amino acids, including this amino acid, this, and that. But remember, it will not tell you which one is the first amino acid, which one is the second, which one is the third, which one is the last, from the N terminal to the C terminal. How are they arranged? That will not tell you. You have to do what we call peptide sequencing. Now discussing peptide sequencing. Peptide sequencing, we have two methods. We have a Sanger sequencing and we have Edman degradation. Sanger sequencing was first carried out by Frederick Sanger, who sequenced hormone insulin in 1953. I think if you can remember, this is the same year. What do you call this person that deduced the double DNA double helix, Francis Crick. This is the same year Francis Crick deduced the DNA double helix structure. So what does Sanger do? Sanger uses a reagent called 1-fluoro-2,4-dinitrobenzene to level and identify amino terminal residue. What did he do? So this reagent, he formulated this, and this reagent can react with the N-terminal amino acid. When it reacts with the N-terminal amino acid, you can hydrolyze. You can hydrolyze after Stella, can you mute?
Oh, sorry, sir. I'm sorry.
Good. So this compound can react with amino acid. Once it reacts with amino acid, usually the amino terminal residue. So once it reacts with the amino terminal residue, you can break down the whole protein. That same reagent attached to the N-terminal amino acid will remain attached to it. So you can isolate it and identify which amino acid is that. The problem with Sanger sequencing is that the Sanger sequencing can only identify the N-terminal amino acid. When it identifies the N-terminal amino acid, this is the N-terminal amino acid. What is the next? Which one is the next one? It is difficult at once. So apart from that, you can also identify the number of peptide sequences in amino acid. You can, I didn't talk about it when I was talking about the structure of protein, being the fact that we will discuss it shortly. But you can have a protein, an enzyme, a functional protein that have two peptides attached together. Two peptides. They are not one peptide. They are two, maybe attached by what we call a disulfide bridge, which I think I already told you what is a disulfide bridge. A bond that is formed between a sulfur and another sulfur. So when you have a sulfur in one peptide and a sulfur in another peptide, these two sulfur in cysteine amino acid, they can form a disulfide bond, thereby attaching the two peptides together to give a one protein. So when you have a protein that contains these two peptides, that protein can have two N-terminal, the N-terminal for one peptide and the N-terminal for another peptide. So you can use this to determine how many peptide sequence you have. So when you subjected this kind of protein to Sanger sequencing, you definitely are going to have two derivatives. Your compound 1-fluoro-2,4-dinitrobenzene will react to two different amino acids. So when you remove, isolate the derivative amino acid and identify, you discover that you have two amino acids, which just tell you that the protein you are working with contain a two peptide, probably linked by a disulfide. And if you continue, you can identify the position of this thing that give rise to the formation of the peptide. We'll see shortly how that can be done. Now, the next thing is Edman. Are we together? So another method called Edman degradation, it also uses a chemical called phenyl isothiocyanate. So this chemical also reacts with amino terminal residue at an alkaline pH to produce, I think I need to show you this. So to produce another derivative called phenyl thiocarbamyl adduct. So this adduct, in the presence of anhydrous trifluoroacetic acid, the peptide bond holding the N-terminal and the remaining will be cut. So in this case, unlike in the case of Sanger's quinine, where all the peptide will be broken, only the peptide holding the derivatized amino acid, the first N-terminal amino acid that react with this reagent, will now be clipped. So once it is clipped, it can be isolated in form of what we call, you can see it here, aniluthiazolinone, derivative of the amino acid. So you can have this being converted under acidic condition to phenyl thiohydantoin, and this residue can be determined using you can determine it using chromatography. So what you have is the remaining peptide, but one amino acid, which is the N-terminal, being removed. So you can continue. You can add another phenyl isothiocyanate, which is the Edman reagent. Again, the reagent can react with the N-terminal amino acid, following the sequence of reaction that lead to the cleavage of that N-terminal amino acid, identifying which amino acid is that, and then add another phenyl isothiocyanate like this. The cycle continues until when you determine all the amino acid. So when you determine all the amino acid in this way, you yes, this is the N-terminal amino acid followed by this one to the last. So when you follow this method, you can be able to identify clearly which amino acid which amino acid is found in this protein and in which sequence do they exist. The only problem with this method is that it can only identify just few of the amino acid sequence. It cannot identify a very long polypeptide. Now, if you want to identify a very long polypeptide, how do you do? Okay, for you to do that, are we together? Are we together? Class, are we together?
Yes, sir.
Aisha. Aisha, are you with us?
Yes, sir.
Okay, so by now we've seen how we can determine the sequence of amino acid, right? A peptide. Now the next thing is, when you have a larger peptide, how do you determine the sequence? It is very simple. Now, when you have, remember you can do that, you can do Edman degradation for a very shorter peptide, like 50 to 70, etc., up to 100 somehow. But when you have peptide containing many amino acids, hundreds to thousands of amino acids, how do you? First of all, what you need to do is you break them into a smaller fragment. But there is possibility that what you are working with is a protein that contains more than one polypeptide. So more than one peptide can linked by what we call a disulfide. So first of all, you have to break the disulfide if in case there is. So just like we said, you can even use Sanger sequencing to determine if it contains the two peptide. It will give you. So once you have an idea and you learn that yes, there is a sulfide bridge, and that sulfide bridge can interfere with your adenine degradation. So what you do, the first thing is to break the sulfide. So there is two ways you can break the sulfide. You can oxidize the cysteine using papomic acid to produce cystic acid. So you can see how papomic acid can be used. We just added papomic acid, which will oxidize the cysteine residue in the disulfide cysteine to form cystic acid. How you can reduce using dithiotretol. When you reduce, you can put an acetyl group so that they will not go back. By adding iodoacetate. When you put iodoacetate, then it will be acetylated. So you will have acetylated cysteine residue. So this acetylated cysteine residue cannot go back to form the sulfide bridge. So by that, you are sure that you've broken the disulfide bond. What next you need to do, you can even use electrophoresis to separate the two peptide. When you separate the two peptide, and then you can continue separately, like each one in a different listing. Now the next step is when you have the peptide. Let's say you have the two peptide and you separated them in a different test tube. So each one, let's just pick one and see how we determine, and you do the same thing for the other one. Now you have one. What do you do next? Like I said, you fragment it into a smaller peptide fragment. How do you do that? You can use an enzyme or use a chemical. You have an enzyme that can clip the peptide at a particular amino acid. For example, you can have an enzyme like trypsin. Trypsin can cut the carboxyl group or carboxy terminal of any lysine residue. So you have a peptide that contains up to 50 peptide, for example, and you have like 4 lysine inside, 4 lysine residue. So it will go to the carboxyl group of this lysine and cut it. So you have 4 from lysine, but it can also cut the carboxyl group of arginine. So assuming you have only 1 arginine, it cuts, then in addition to that, you ended up having like 5 fragments altogether when you use trypsin. So you may decide to use another one like chymotrypsin, which cut the carboxyl group of phenylalanine, tryptophan, and tyrosine. You can use pepsin, which cuts the N-terminal in this case, N-terminal group of phenylalanine, tryptophan, and tyrosine. You can also use a chemical like cyanogen bromide, which cuts the carboxyl group of methionine only. You can see a list of some of these reagents. All of them are enzyme with the exception of cyanogen bromide. So you can use any one of them. You may decide to use one. But we will see how using 2 is important. But you don't use 2 all in one test tube. So you have just the peptide and you put it in you take the sample into 2 different test tubes. Each of the test tube, for example, you said, okay, this one I will treat it with trypsin. This one I will treat it with cyanogen bromide. So you continue. You see how important it is to do that. Now the next thing is you sequence it using adenine degradation. So when you have the fragment, you can after using for example trypsin, you put trypsin and you have 4 fragments in by treating with trypsin. So this fragment, they are smaller fragments. You can sequence it using Edman degradation. So when you sequence it using Edman degradation, you are going to have post-sequence. When you have post-sequence, the next thing now is ordering and overlapping. How do you, ordering the overlapping sequence? How do you know that this sequence occur in this order? That this is the first one, followed by this one, linked by this one, and you have your own peptide. How do you do that? Now, this is where the point of having two different test tubes containing the same peptide, but you fragment it using a different chemical. For example, the first one we said trypsin. Well, the second one, you decide to say, okay, let me use a cyanogen bromide. And when you do with cyanogen bromide, it gives you, for example, 3 peptides fragment. So you also sequence them. Of course, it's the same protein. The only thing is the different fragment. This one is 4 fragment, while this one is 3 fragment. So how do you arrange them? You now look for the overlapping sequence. How do you look for the overlapping sequence? You look at the fragment produced by cyanogen bromide and the fragment produced by trypsin. Of course, they will not be the same fragment. This one produced 3 fragments, this one produced 4 fragments. So definitely when you look at the sequence, you will see an overlapping. For example, let's say, look at the one you have, just imagine this thing I'm saying. You have the same amino acid sequence. I don't know of any amino acid with O, but look at the sentence I have on this, well, let's say press on this, the title of this slide, say ordering the overlapping sequence. Let's say ORDERINGTHE, like this, up to E of sequence. We have that as the sequence of the amino acid, the overall protein. So we have ordering. When we use cyanogen, when we use trypsin, the trypsin cuts ordering, the overlapping sequence, and you have this sequence. You have this 3-4 fragment. And when you use cyanogen bromide, the cyanogen bromide decide to cut order, just order without ing. It cut order without ing. And then then the next fragment contain the ing, v, over. Then you have ing, v, over as one fragment also. Then the next one, you have lapping sequence at the last one. Class, are you with me?
Yes, sir.
Yes, sir.
Are you really following?
Yes, sir.
So when you have this fragment now, how do you arrange that, or how do you deduce that the sequence are in this format or are in this way? What you would do is you look at the first fragment. For example, the first one produced by trypsin, say ordering. You have ORDERING in this sequence. And when you look at the one produced by cyanogen bromide, you see ORDER only. You say, okay, so this fragment that started with ING, or bar is linked to the ordering, the order, the first one that had order. So you can link this one, so you have ordering the over. Then you look at the next sequence produced by trypsin. You have overlapping. That will give you an idea that, okay, so this lapping sequence is now attached to this one that stopped at over. Now you have overlapping sequence. You ended up with the whole sequence. Is that clear? Class?
Yes, sir.
Is that clear? Now, from this fragment, you can be able to, no matter how long your sequence, you can be able to identify the exact sequence, even if they are in thousands. So this is basically how you determine. Now, we talked about structural organization of a protein. Now, how do you, remember I told you that when you have the amino acid sequence just attached by the peptide bond, we call it primary structure, right? When you have the amino acid sequence attached to each other, we call it a primary structure. Plus, are we together?
Yes, sir.
Yes, sir.
Yes, sir.
There is this thing I did not include. That's why I'm a bit surprised. I talked about locating the disulfide band. Now, after having your sequence, and you also do the same thing for the other sequence, when assuming, say, you have a protein that contains two peptides, how do you determine where is the disulfide band? So you do the same thing, you get the sequence for all of them. What you need to do now to determine where the disulfide exists is, before you take your sample now, the fresh protein sample, where you didn't remove or you didn't oxidize the disulfide bridge, now contain your disulfide bridge and say, okay, let me treat this with cyanogen bromide. So when you treat with cyanogen bromide, you will know how many fragments you will obtain when you run it on a gel electrophoresis. Because you have done it when you break the disulfide and you treated with, for example, cyanogen bromide, you run electrophoresis, you have fragments, and you take all these fragments and sequence them. Now you do it without removing the disulfide bridge. So of course, that location, that point where it contains the disulfide bridge, you have the two peptides, these two fragments will be together. So when you run the electrophoresis, you will not see those peptides. All of the other peptides, you will see them on your gel, but minus these peptides, which only tell you that, okay, this peptide and the other peptide that are not appearing in their initial, but I can see one bigger peptide here, which formed a new one, is because of the two of them, they were linked by a disulfide bridge. So you already know which peptide are this, and you can also, because you pick the peptide and sequence them, you know which sequence are they. Then from that sequence, which of them contain cysteine, you definitely see cysteine in all of them. So it simply says it is between this cysteine and the other cysteine residue that the disulfide bond is formed. So from there, you can be able to identify where the disulfide bond exists. So talking about the structure of structural organization of protein, we told you that the protein is organized in a way that it can perform the function. First of all, when you have the amino acid linked by the peptide bond, just the amino acid by the peptide bond, we have what we call the primary structure. So this is just because of the peptide bond linkage. But there are quite a number of interactions that could happen between the amino acid, apart from the peptide bond. There could be some steric relationship within the linear sequence. And that steric relationship can be in form of hydrogen bond. Maybe the first amino acid, apart from the peptide, can also involve in a hydrogen bond with the next amino acid, and that one also with another amino acid like this, like this, like this. That leads to what we call a secondary structure. So that secondary structure, because of that interaction, it leads to formation of secondary structure. So these secondary structures are in two forms. There is alpha helix and there is beta sheet or beta pleated sheet. I think I will explain it better here. So in alpha helix, what happened in alpha helix is you have the amino acid sequence, like let's say 10 amino acids. The first one is serine. There is possibility of serine forming a hydrogen bond with at least not the second, the one next to it, with at least the fourth one. That one can also form a hydrogen bond with the fourth one. So you have the carbonyl carbon of serine, and let's say the amino, the nitrogen of the next amino acid, of the fourth amino acid forming a hydrogen bond. That one also is on carboxyl, the carbonyl group, and then the nitrogen of the amino group of the next, which is supposed to be, we say fourth, the next one will be 8, like this, the next one will be 12, the next one will be 16, like this, like this, like this, and they will be forming a hydrogen bond. And that forming hydrogen bond leads to twisting of the whole structure to what we call helical structure. And that gives this helical structure we can see. So the twisting can lead to formation of the helical structure. And this helical structure is the most common form of protein or peptide secondary structure. It can be right-handed, it can be left-handed, but only right-handed proteins are found in living organisms. Even though you can have some other group, acidic amino acid and basic amino acid that can actually interrupt this structure because of the additional group that are charged. For example, in the glutamate, you have the carboxyl, additional carboxyl group that has a negative charge. Is something in aspartic acid, like in basic amino acid, you have additional positive charge in lysine, histidine, arginine, etc. So they can interrupt that, but basically, all of them can involve in this hydrogen bond, which leads to formation of helical structure. But in a situation, the hydrogen bond does not form between the amino acid within a sequence. When you are talking about amino acid sequence, maybe of 2 peptides. So you have the hydrogen bond formed between the carbonyl of one peptide and then the amino nitrogen of the other peptide, forming this hydrogen bond like this. It leads to formation of the beta pleated sheet or beta sheet. Or in a situation where you have just like here, you can see when you look at this primary structure, you can see how this amino acid sequence pull back on itself. So when it pull back on itself, hydrogen bond can